Boogie. Today I'm talking about transcription. This is part 2. So we have our pre-mRNA here, RNA, and so this is going to be the 3' end, this is going to be the 5' end. And so what's going to happen is now that we have this pre-mRNA, we need to get it packaged for mRNA. So you're going to have sections that are called interons and exons. I'll call this an exon. This might be an interon, and then an exon, and then an interon. And what you're going to do is there's a thing called an enzyme called a spliceosome, and that's going to cut out all the interons, and you leave the exons. So it's kind of backward. The exons stay, and the interons leave. And based on the order that we move the interons, so maybe we have an exon here and an exon here, and so we take these two exons and we switch the order of them, we're going to be able to have a lot more genes labeled on one specific DNA. So the arrangement of these little splices put together can give us different proteins as well, so the DNA stores a lot. And then to finish off our mRNA, we're going to need to put a methyl cap on. So we put on our methyl cap on the 5' end. Put a little star by that on the 5' end. So I'm drawing a little hexagon, coloring it in. There's our methyl cap. And then on the 3' end, you're going to have a poly A tail. So a poly A tail on the 3' end. A. And these two, the caps, help protect the mRNA outside of the nucleus. mRNA outside. And so when the strand of mRNA is going to leave out through a nuclear pore, and there are enzymes out here, so when the DNA goes to the ribosome and then it gets translated into the protein chain, then you have this mRNA sequence and it needs to be broken down. And so these enzymes will come along and break it down. So the poly A tail and the methyl cap keep this strand from being broken down before it is translated into protein. And I hope that was helpful.